Hello, my friends. Thank you so much for joining me. My name is Laide Akinfadeni, and welcome to my case study Pharm001B. And here I will be looking at a theoretical or two theoretical programs, LAT101 and LAT202, and I will be looking at them as and using them as an example to apply, if you like, the knowledge that I have gained from various training platforms, various technologies, and various journals that I've read, and that I can apply to an actual theoretical product. So this case study will provide an overview of the life cycle of CAR T-cell therapies. So from product conception, product development, preclinical studies, through to clinical trials and investigational new drug applications and marketing authorizations and biological license applications. So it's going to be an overview of the whole life cycle activities, and it will all be captured in a life cycle management manual, and that will be what I will be using in another case study. So these programs, they are theoretical. They're not actual real products. They are theoretical products that I've just put together. But they are based on contemporary scientific advances, as I've stated in all of my presentations and will state. And they're just going to be looking at how CAR T-cell therapies can be used to treat cancers, including hematological malignancies and solid tumours. So I'll be taking you through a series of milestones for these programs, and we will be looking at the regulatory pathways. We will be looking at the products itself, the immunomodulating drugs, and we'll just be looking at all of the considerations and applying all of the knowledge that I've gained from these courses and from these platforms and from these technologies, and consolidating it into this project. So this is my quote for this presentation, and the quote is that innovation is the ability to see change as an opportunity, not a threat. And it's really important that when things happen, and that you have a good mindset. And I feel that I have learned a lot in these past 3 to 6 months, more than I've learned in the 21 months that I was in another environment. I learned so much, and I think it has been an opportunity to learn and to innovate and to become resourceful, and to see these changes that have been enforced upon me, but to see it and to use it as an opportunity to better myself, and not to see it as a threat, and not to see it as something that will demoralize me, but to use it and to use the time and to use the resources that are available to me in order to better myself and make myself a stronger person, and just for my own sake and for my own enjoyment and for my own knowledge, because obviously this is my own project. It's not going to be used for anything. And many apologies. I had originally in the an overview of the process, but it's flipped through that slide and it's gone onto this slide. I don't know why it's done that, but it doesn't matter. We don't need to go through the overview of the presentation. This is a cartoon-like diagram or illustration of LAT101, and it is a tandem CAR T cell. So it's a bispecific, i.e. the antigen recognition recognizes two specific separate antigen or molecules. So it's the CD19 and the CD22, and these two molecules are specific to B cells. So this is a product that is going to be targeting B cell malignancies, and so the antigen recognition domain is going to be targeting the two products that are specific to those B cells. So let's just look at the illustration just to gain an understanding as to how the CAR is constructed. So the CD3 is derived from the CD3 complex. So the CD3 complex in normal T cell receptors is bound, non-covalently bound to the T cell receptor, and the CD3 zeta is the part that's responsible for the activation. So the CD3 complex consists of the CD3 zeta, CD3 gamma, delta, epsilon. Yeah, but it's the CD3 zeta part of that component that's been constructed in order to use for the ITAM part, the I think it's immunotyrosine based activation motive for the activation of these T cells. The CD28 is the co-stimulatory domain that we've selected for this particular construct, and the transmembrane domain is the CD28 because it's compatibility, and this is bound to a hinge, and then we have the antigen recognition binding sites, or the antigen recognition domain. So it's CD19 and CD22. So this will be delivered by a lentiviral vector, and it will be delivered ex vivo to the patient's cells, which are the starting material. So this is just, as I said, an illustration, a cartoon illustration. In reality, all of these T cell receptors are covered. The T cell is covered with T cell receptors, CAR receptors, and other T cell receptors as well as other receptors that are and molecules that are specific to the T cell. So the CD8 may be present, and the CD4 plus may be present, as well as other molecules like or other receptors and molecules. So this is just, as I say, a picture to represent it. So this is the second program, LAT202, and as I said, this is looking at two CARs. So it's not just one CAR, we've got two separate CARs. This is going to be by gene editing, and the gene editing will be the CRISPR-Cas9 editing tools, and we will insert one of the CARs into the TRAC, the TRAC, the T cell receptor alpha constant region, and the other CAR will be inserted into the a safe harbor position. But this is the design that I've come up with. So for the for the HER2 and MUC1, these are the antigen recognition domains. The activation signal will come from the CD3 zeta, which is derived from the CD3 complex. The co-stimulatory domain, each CAR will have a different co-stimulatory domain. So the MUC1 antigen recognition CAR will be connected to a CD28 for co-stimulation, and the HER2 will be 41BB. Now, the co-stimulatory domain is responsible for the cytokine profile that dictates the type of phenotypes. There's the phenotypes that are produced on proliferation, and the phenotypes that are produced on activation. So the type of activation and the spread and the type of T cells that will be generated, and hopefully you will get that persistence, that long-term persistence and long-term cancer or tumor killing effect. That's the purpose of using this dual approach. So this is the second program, and I hope that, oh yes, I wanted to talk about the transmembrane domain CD8 alpha. So CD8 alpha is derived from the CD8 co-receptor. So the CD8 molecule consists of two parts. It's a heterodimer. It consists of the alpha region and the beta region, and it's the CD8 alpha region that's been used to construct this transmembrane domain. For the CD28 co-stimulatory domain, the transmembrane domain selected is the CD28, obviously for compatibility reasons, which is also selected as the spacer or hinge that is bound to the antigen recognition domain. So this is going to be manufactured using induced pluripotent stem cells, and these induced pluripotent stem cells will be gene edited using the CRISPR-Cas9 editing tool, and then they will be differentiated, and master cell banks and working cell banks will be produced. So that's kind of the strategy that we're going to be going for. So this is going to be an off-the-shelf product. It will be genetically, or so it will be gene edited in order to reduce the graft versus host and the immunogenicity effects that you will have with allogeneic products. So some of the HLA genes will be knocked out in order to prevent that rejection of the T cells that will make up this product. So this is just a slide giving you a description of LAT101. So it's an autologous CAR T cell therapy generated by ex vivo transduction, and it's designed as an OR-gate tandem CAR targeting CD19 and CD22. So what that means is basically this product can target either CD19 or it can target CD22. You will get activation if the antigen recognition domain binds with either one of those targets. The product is engineered with a co-stimulatory domain of CD28, and it also includes an inducible caspase 9 safety switch, which is introduced via gene editing technology. That's quite clear. You want to have that safety switch to protect against adverse reactions that you could potentially normally get with regards to cytokine storms, so cytokine, CRS, and neurotoxicity. The product is, so I think I said that, LAT101 is bispecific, designed to activate T cells upon recognition of either of the antigens and reducing the risk of antigen escape. And it's intended for the treatment of relapse of refractory B-cell malignancies, including diffuse large B-cell lymphoma and acute lymphoblastic leukemia. So the product LAT202 is the second program, and this program is, let me just close the window. LAT202 is an allogeneic product. It's going to be an off-the-shelf product, and it's going to be primarily manufactured by gene editing using the CRISPR-Cas9 gene editing tool. It will consist of an AND gate bispecific targeting mechanism. What that means is that both of the antigens recognition domain have to be bound. It has to bind to each of its antigen in order to produce that activation of your T cells, and that makes this product a little more precise. And the reason being is that HER2 the human epidermal growth factor receptor 2, is expressed on healthy cells. However, for tumour cells, you do get an overexpression of antigens. Where there could be there's a number of antigens that are expressed, and you do get this overexpression, and that's one of the characteristics, if you like, of tumour, is that you get this overexpression. The product comprises of 2 distinct CARs, each with one co-stimulatory domain, CD28 on MUC1 targeting CAR and 401BB on HER2 targeting CAR. And that's to improve what am I thinking of? Is to improve the cytokine profile, to get us a broad profile so that you can get a broad phenotype typical T cells that are produced as a result of this activation and proliferation. So we want to have that cytokine profile to be able to produce in vivo on activation, as well as ex vivo. You want to be able to produce a range of phenotypes, and so therefore I've gone for this dual co-stimulatory domain, CD28 and 401BB. LAT202 T cells are further CRISPR-Cas9 engineered to resist TGF-beta mediated immune suppression. So this is the tumor microenvironment, and the tumor microenvironment consists of other components as well as the transforming growth factor beta ligand. So it also consists of the macrophages, M1 to M2, and other factors that contribute to the tumor microenvironment. But this is one way that these cells are being designed in order to resist the tumor evasion through this tumor microenvironment by engineering the TGF-beta receptors. I don't know whether that's quite clear there, but it's the TGF-beta receptors that are engineered such that they don't inhibit T cell in activation, but they actually activate the T cells on binding with the TGF-beta ligand. So it's a way of flipping the normal response that normally happens where you have this TGF-beta mediated activity, if you like, but what the tumor microenvironment is exploiting in order to evade immune killing, and or to evade the immune system. So because of the fact that this LAT202 has been engineered to resist that and to flip that particular scenario, so instead of causing inhibition, it causes activation, or it causes co-stimulation. It means that the T cells can actually get to the tumor cells, and they can actually be effective and efficacious. Additional engineering features includes the inducible caspase-9 safety switch. Again, that's to prevent or to be able to deal with severe side effects and immune responses, such as neurotoxicity, but also it's going to have the expression of the chemokine receptor CXCR2, which enhances tumor-directed trafficking. So LAT202 is intended for the treatment of advanced or metastatic solid tumor co-expressing HER2 and aberrantly glycosylated MUC1. Such as pancreatic, lung and breast cancer. And the AND gate logic ensures that the T cell activation only occurs upon simultaneously recognition of both tumor associated antigen, thereby improving tumor specific specificity and minimizing off tumor toxicity. So basically we want to reduce the on target off tumor effects, toxicity effects, because of the fact that HER2 is expressed on normal healthy tissues. However, MUC1 is expressed on, or this aberrantly glycosylated MUC1 is expressed on tumor cells, and it is a tumor specific antigen. So remember, the MUC1 ordinarily is highly glycosylated, is highly O-glycosylated with carbohydrates, and so you don't get any exposure of the peptides. But because of the tumor and the mutations that occur, you get this truncation, and you get the revealing, if you like, of some of the peptide residues, where an epitope can be identified or has been identified and utilized in order to attack or target these cells. Right, so I'm going to talk about the target product profile for each of these products. So LAT101 is an autologous product. It is manufactured via ex vivo lentiviral transduction. It's autologous, which means that the patient who donates the sample will be receiving the product that is the patient that donates the starting material, that starting material is used for his the final product that is given to the same patient. So it's important that we have mechanisms and we have logistics in place to check for the to ensure that the patient receives the correct actual medicine, because this is a personalized medicine and we don't want to be giving the patient medicine that belongs to some other patient. So we have to incorporate a chain of identity, and within that chain of identity will include some analytical testing, genetic testing, to confirm that the right patient is getting the right drug. So this will be short tandem repeat analytical testing to confirm, and this is part of regulatory requirements, to confirm that the patient who has donated the sample, the starting material used to manufacture and make the product, is receiving the actual final drug that has been manufactured from his or her sample. The testing also confirms that there's been no contamination of other components as well. It's just simply the patient. So it is part of the chain of custody documentation that is required for autologous testing. The other thing to take into consideration is the indication, who's going to be getting these drugs, and it's mainly going to be for it's for the treatment of relapse and refractory B-cell malignancies. It is going to be for patients who don't have many limited options, if you like, and where there is an unmet need. We do need to be mindful if patients have already received immunomodulating drugs. If they've already received CAR T cells, we need to be mindful of the differences between the samples, the starting material donated by patients who have already perhaps received CAR T cells and perhaps will be receiving this particular drug, and patients who haven't. So when we're doing our clinical data analysis, we need to just be mindful of what we're comparing against. Are we comparing against patients who have a starting material that may be significantly or not different from patients who have never received CAR T cell therapies? The patient population is going to be adults and pediatric patients with CD19 plus or CD22 plus B-cell cancers. So we have to test the patient to ensure that they do have those particular, that they do have those particular, what we're calling antigens present on their B-cell cancers. And we want this patient population, which would be part of our clinical trials, it aligns with the concept and the strategy for high risk and therefore a high need group. So it's going to be scientifically driven. It's going to be given to patients where there are limited options because of the fact to give it to patients where they have other options where we know the safety and therapeutic profile is more of a risk, and we have to measure the benefit and of the drugs against the risks associated with giving this drug to patients who perhaps have other options. So the patient population will be high need groups where there are limited options because of the risks associated with this product, because it's a development product. The mechanism of action is due to antigen recognition, T cell activation, proliferation, and the targeted cytotoxicity against the malignant B cells. And the incorporated IC9 inducible caspase 9 safety switch in case of cytokine storms, uncontrollable cytokine storms. The dosing will be a single IV infusion of autologous CAR T cells after lymphodepletion. This is your normal standard practice for these types of products. The dose regimen, well, I've based this on what is currently available, but we will see. It may be updated with time and with more data. So we will see. It's likely to be a single. Very unlikely to be repeat doses. This is meant to be a single dose approach. If we are doing any potential for repeat dose, then we need to make sure that our non-clinical studies also could take that into consideration. So if we are going to ever be in a position where we're going to say yes, for this particular situation or this particular scenario, we're going to give another dose, which, bearing in mind, thinking about the science of it, if you're given another dose, does that mean that you're going to have the same sample that was used in the original dose, or are you going to take a new sample from the patient? I think you would be using the original sample, but we don't know. But in any case, all of those have to be supported by non-clinical studies. So the non-clinical studies have to mirror what is going to be done on your clinical trials. And your efficacy endpoints, so these are, what is already available in biological license application and MAAs for CAR T therapies. It's just basically the industry standard endpoints. ORR stands for overall response rate. CR stands for complete response. PFS, progression-free survival. OS is the overall survival, and MRD is the minimal residual disease negativity. So it should be 0. No disease should be detected after ideally you'd want it to be 0. And we'll just go through the regulatory pathways in more detail in the next milestone. So this is the target product profile again for LAT101, and these are what we kind of expect, what's current in the industry, current industry practice. So we understand what the adverse reactions are likely to be, the neurotoxicity, and we understand and we have an approach in the industry as to how we're going to deal with that. We do need to have a way of grading and reporting the levels of neurotoxicity, but we understand what the expected side effects are going to be, and we have a worst case scenario as to how to deal with that. Storage and handling, cryopreserved cells. So we have to generate data to justify not just the fact that we can store it for 12 to 24 months, if that's going to be the case, if we're going to freeze, cryopreserve these cells, but how do we release them? We have to look at the cell viability. We've got to look at the compatibility of the cryopreserving agent. We have to look at the in-use stability and in-use compatible with other diluents that may be used to administer the drug, or other equipments and containers and that can be used to deliver the drugs. So storage and handling we've got the minimum information there, but we do know that we have to do a lot more for that. Manufacturing platform is going to be autologous lentiviral vector gene transfer. It's going to be a closed manufacturing, closed system for manufacturing. We'll be operating to current good manufacturing practice. What makes this product different is the dual antigen approach. The safety switch is something that's already ongoing in other clinical trials, so it's not really different. But if we're the first to market, then it could be. And also we will use data from what we've already got available. So platform data and prior knowledge data will be made available. But the dual antigen targeting, what makes that effective or better, and what differentiates it, is that it mitigates for cancer escape. So sometimes you may get CD19 underexpression, and then those cells proliferate, and those cells become dominant, and we have no way of accessing them because they're not expressed. So we have another target, the CD22, to aim for. So this is a way that we mitigate cancer cell escape. And the regulatory pathways, these are standard EU, standard UK's, and standard US pathways that are available. And the objective is to ensure that we have that regulatory engagement throughout, right from conception right through to the development, early development, prior to non-clinical studies, non-clinical studies, clinical studies, and post and marketing authorization and post-approval. So we really want to have, throughout the whole life cycle of the product, we want to have that early engagement with the regulators, and we want to take advantage of schemes such as ILAP, such as Interact for the US, such as the Innovation Task Force and Prime designations. All these mechanisms that are and frameworks that are available that countries have put up in order to ensure that we get this engagement, so that we have drugs that are successful because we've done what the regulators expect us to do. This is LAT202. So LAT202 is going to be an off-the-shelf product. So it will have certain not characteristics that are different to the personalized approach for LAT101, and those are that we want to be avoiding the rejection, immunogenicity, as a result of the fact that this product is allogeneic, so therefore the donor and the recipient are going to be different. So the product is going to be gene edited in order to reduce that rejection, that potential for rejection. So we will be knocking out some of the HLA genes, the human leukocyte antigen genes, A, B, and D, I believe, and we will be knocking in maybe I think it's G and F. I can't remember which ones. We'll be knocking in human leukocyte antigens, and the whole purpose of doing that is to reduce the rejection of the product by the body's immune system, and to prevent the attack of the cells by the natural killer cells as well. So there are certain advantages with using an off-the-shelf product, and that would be the time that we can give the drug to the patient, and that's what's a differentiator for this. However, there are unknown quantities that need to be taken into consideration, and so therefore, because there are some unknown and some potential side effects that we can't predict. This needs to be given to until we have a lot more data and experience with it, where there is a high unmet need in solid tumor. So we have to consider all of the unintentional effects that could occur from gene editing, and those can be the unintentional effects that could occur from on-target gene editing, so where we edit the genome of the inducible pluripotent stem cells, where we edit the genome in the correct place, and off-target where we perhaps make gene edits in places that are not where we predict or where we would want them to be. So we have to look at the potential for that, and we have to validate those systems. We have to validate the gene editing components. There's a different emphasis. The emphasis is on the control strategy for your gene edited components, and in the FDA recommends that the gene editing components be considered as a separate dossier, so a separate drug substance dossier for your gene editing components. So we have to really consider what kind of patient population is going to be receiving these products, and where there are already medicines that are available. Because of the unknown risks associated with this, we would suggest to patients who have options to use those medicines. But where there is a high unmet need for patients who have few or no options, this would be a suitable candidate if their tumour has been identified as being the overexpression of HER2 plus and MUC1 plus antigens. So this is a really good or well designed, I believe, product, because we're thinking about the potential roadblocks, the potential problems associated with dealing with tumours, and that is the tumour microenvironment. So the TGF-beta resistance is something that has been challenged by engineering the receptors, the TGF-beta receptors, so that once it binds with the ligand, the TGF-beta ligand, that instead of having an inhibitory effect, it has an activating or co-stimulatory effect. We also have the inducible caspase 9 safety switch. We also have the CXCR2 for tumor trafficking, and that's to guide these cells to the tumor and to reduce the off-target, the off-tumor toxicities. Dosing will be single IV infusion. If we need to do repeat dosing, it is possible because we have an off-the-shelf product. The cost of producing this product is going to be significantly less. And it's just going to be a much more convenient dose. For example, for autologous products, the patient, there's a vein-to-vein time, delayed or lag time from when the patient gives the sample to when the patient receives the product. And the problem with that is if we're dealing with a particularly aggressive tumour, then or cancer, then we may find ourselves in a situation where there's a patient who has this patient has no longer meets the eligibility criteria at the time when the drug product is required for them, because of the fact that they have this aggressive tumour. And if we do give the patient some drug to treat the tumour in the intermediate time, when we have to take that into consideration when we're analysing our clinical data to see whether or not the effects of the drug was due to the actual drug or due to the actual drug that was administered to the patient in the intervening time whilst we were manufacturing our product. So, we have to really think about who we're going to be utilizing or who's going to be participating in this particular trial, and we have to get the patient's buy-in to this approach. And also the starting material, I don't know whether I said this for the LAT101, it applies to LAT101 as well as LAT202. If patients have already had relapse or refractory cancers or tumours, say, then if they have received a CAR T cell therapy, then their starting material is going to be different. This is for LAT101, so there may be potential differences. But even for LAT202, we have to take into consideration, or we have to consider, whether or not those patients have received any kind of CAR T cell therapy prior to our particular drug, and that might be where we have to take that into consideration in our data analysis of the clinical data. The mechanism of action, dual specific, so it's targeting HER2 plus and MUC1 plus antigens. TGF beta resistance, I've talked about that. Dosing, single IV infusion, but the repeat dosing is possible because this is an allogeneic setting. At the moment, we have chosen this dose based upon what's already out there, and the efficacy endpoint, again, this is reflected in solid tumour clinical trial standards. So we have the same kind of sort of adverse events with regards to neurotoxicity and cytokine release syndrome, but we also have this graft versus host disease risk, and what we've done by mitigating that risk is we have knocked out some of the genes that can potentially be allow the recipient's body to view the product as foreign and therefore attack the product. So we've knocked out some of the genes, we've knocked out the T cell receptors, and we've replaced it with the CAR gene. So that now means that the T cell receptors do not identify the recipient's body or the recipient's cells and tissues as foreign and attack them. So you don't get the graft versus host disease because we've knocked out the T cell receptor gene. Or the T cell receptor gene that expresses that T cell receptor has been knocked out, or the T cell receptor alpha constant region has been knocked out. The same comments that I've raised applies with regards to storage and handling. These have to be validated, so you have to do stability, you have to do compatibility studies, in-use stability studies to validate and qualify the use of these products. It's going to be gene edited, and we're going to be doing a long-term follow-up with these both of these products, with LAT101 and LAT202, because of the fact that we're using gene edited products, and there are high risks associated with gene edited living drugs. So we have to do long-term follow-ups, at least 15 years, according to the FDA. I think it's more in the EU, but I'll double check that. So how is this product differentiated? It's differentiated because it's off the shelf, and it's going to be cheaper. It's going to be easy to access. So if people have aggressive tumours, they'll be able to get hold of the medicine quickly. They don't have to wait for a long time, or they don't have that lag period. And it's a better choice, if you like, for tumours, because it has this enhancement where you get this trafficking, and you also get the counter, or the medicine has been designed, the CAR T cells, the T cells have been designed to mitigate the inhibitory effect of the TGF beta ligand. The regulatory pathways are going to be the standard as described previously, but the most important thing that we need to talk about is that we need to make use of scientific advice and all of the meetings that are available at all stages of development, right through to concept, through to non-clinical, through to preclinical, through to clinical trials and marketing authorization. And because of that early engagement, we can inform the regulators of what we plan to do, in terms of what we plan to do right at the start, if they agree with our strategy, if they agree with our approach with regards to the analytical testing that we may plan to be doing for comparability studies that we will likely need to be doing later on in the development program. So we need to have that early engagement, and we need to have that those discussions with the regulators. I think I've already discussed this, but this is something from a company perspective. If we talk about this fictitious company, LAF Therapeutics Limited, what we need to do is we need to consider the vein-to-vein time for LAT101. At the start of the patients who may be receiving these drugs, they will may have aggressive forms of cancer, which by the time the manufactured material is available, they may be ruled as ineligible for the use of this drug. So we have to take that into consideration. Compatibility, stability, compatibility. I think I've already talked about that. That information has to be in the CMC dossier, CTD dossier, in the CMC information of the CTD dossier. We've got. I mean, I've already discussed this. We need to also monitor the transduction efficiency for LAT101, where we have this tandem domain. We don't have that issue with regards to the gene-edited LAT202, but we do have that issue for LAT101. So we just need to manage the transduction efficiency. Sorry, I didn't put a reference there for LAT101. We also need to consider potential for immunogenicity, basically, where we have the formation of ADA, anti-drug antibodies. So, for example, the binding domain, the antigen recognition domain, may be derived from murine sources. If we do any humanization of the complementary determining regions, if we do any humanization, we need to determine if that has an impact on the antigen binding or the affinity or the affinity of the recognition domains. So we just need to make sure that we are taking steps to minimize immunogenicity, and the steps that we take to minimize immunogenicity doesn't result in any changes that are detrimental to the therapeutic outcomes and the therapeutic ability of the drug. And the other thing, as I said, we need to consider the variability in patient-derived starting material. And it may lead to difference out in regards to transduction, particularly if patients have received immunotherapy drugs. I think I've already discussed that. And this is just an overview of where we're starting at. So the blue boxes are where we're starting at, where I feel that we don't really need to have an ATMP classification and certification, but maybe some countries within the European region may require that. It might be one of their requirements. So we would need to find out. So, for example, if we're intending to go for a clinical trial in, say, Switzerland, which isn't part of the European Union or the European Economic Area, they may require an ATMP classification. But for these kinds of medicines, it's pretty clear in the EU what the classification is, and that it is a cell-based medicinal product. So it's an advanced therapy medicinal product, and it falls under the category of somatic cell-based medicinal product. In the United States, this is a gene therapy product. So because of the fact that both of the LAT101 and LAT202, because of the fact that the gene has been integrated into the T cells, however you've done it, whether you've done it via the lentiviral vector strategy or via the gene editing strategy where you use the CRISPR-Cas9, you have that integration and the change of the genome. So therefore it's a gene therapy medicinal product, and it's and that's the classification. And there are certain guidances that the FDA have put out with regards to their expectations, particularly on gene therapy medicinal products, with regards to the unknown or unintentional long-term effects and the long-term follow-ups, and the unintentional effects as a result of gene editing, and the unintentional effects relating to off-target editing as well. So I think I kind of digressed a little bit, but the point of this slide is that we want to show regular continuous regulatory engagement with the regulators, not just with the countries that you're necessarily planning clinical trials and marketing authorization, but with countries where you're not. What would be their requirements? Would they be what could they accept? What are the good tissue practices that we expect? What are the testing regimens that should be conducted on starting materials that are going to be used for allogeneic drugs? So what are the eligibility requirements? Are we going to be using starting materials that have already been fully characterized? So induced pluripotent stem cells that have been fully characterized, for which regulators have already reviewed and assessed the dossier. We are going to be using CRISPR-Cas9 technologies for which regulators are fully aware of in terms, and so therefore we'll be utilizing prior knowledge. So we have to take all these things into consideration and consolidate all of that information in our communications, in our briefing documents and our communications with the regulators. So this is what would happen if we are doing an ATMP classification. I think I've put down that I did one just for the sake of doing one, but I really do feel that this is a well-known and well-established type of therapy. So it's not really something that really requires ATMP classification or certification. It's pretty obvious what kind of product this is. We would discuss this with our early discussions with the regulators and find out if they do want us to go for this ATMP classification and certification. On first instinct and first principles, it's not really relevant, but however, it's important. One of the important concepts here is that the process is the product, and therefore it's very important and key that we characterize our manufacturing method as quickly as possible. And we want to understand what the critical quality attributes are with regards to and how they're linked to the therapeutic efficacy and safety of the product. So all of these things are important things to consider with regards to ATMP classification, but with regards also to the product itself. Yeah, I think I've already discussed about full characterization, that, as I said, for this particular product, we kind of understand what is going on. We understand it because there's a lot of information out there. There's a lot of clinical studies out there, and we understand that this is a cell-based medicinal product. But if you're having any discussions with the regulators, it's good to have as full a characterized manufacturing method as possible. As much information as possible you need to give them in order for them to be able to give us practical and relevant feedback. But if you can start off with fully characterizing your manufacturing method, and having that available so that you can discuss with them your long-term strategies and goals, and those long-term strategies and goals will include scaling up and scaling out, and what are the potential pitfalls and what are the potential problems that the regulators would have, and how they would want the company to address them, to mitigate them. What would they prefer us to do? Is it worth us spending all this money scaling up and then only for them to reject our applications? So it's important to set the foundations down and to discuss them with the regulators and to find out the kind of information they want earlier on. So what kind of analytical procedures, what kind of testing they want us to perform on our products, and what kind of tests should we get qualified, if not fully validated, in order to be able to make those comparability assessments later on in our development program. So We want to be able to provide the regulators with as much information as possible to inform them of their decision making. All of the manufacturing plants and the manufacturing sites must operate to GMP, current good manufacturing practice. Non-clinical programs are used to inform your clinical trials and your clinical strategies. Your non-clinical data should, as much as possible, mirror what we're planning to do in the clinic in the human models. Your non-clinical programs have to be predictive of your human responses. If you are deciding to use a surrogate approach, to include a surrogate approach, so for example, you know it's not reasonable to use the human donated product that has been on in murines or in mouse because of immunogenic response, and therefore you would have a surrogate approach where you would use T cells that are derived from the murine sources themselves to check and to look at the long-term effects, to look at the dosing, to look at the dose to be applied, to look at the administration, etc., etc. If you're going to be doing repeat dosing in humans, you must do repeat dosing in the murines in your non-clinical studies. So your non-clinical studies have to, as much as possible, reflect what you're planning to do in the clinic. They must be able to provide you information on safety, on efficacy, on toxicity, and on drug administration as well as dosing. So all of these things are important, and before we spend a lot of money on in vitro studies, on in silico studies, and on in vivo studies, which are very expensive, we need to we need to get buy-in from the regulators with regards to the use of the animals that we've selected, the use of the 3 R's to reduce, replace, or what's the other one? Reduce, replace. Can't think what the third one is. But anyway, we have to have their buy-in, and we have to get their agreement that before we spend a lot of money on non-clinical studies that perhaps may not be sufficient to support our clinical trial. And prior knowledge, whatever information is already out there, we don't want to reinvent the wheel. We'll just simply refer to them and include them in our literature references. And then I have this life cycle management manual. So this life cycle management manual is going to be a living document. It's going to be there in order to ensure that all of the relevant departments know what the past is, know the history, know where we are now, the status that we're currently in, and understand where we're going, not just from each other's particular group, but from other aspects, from other stakeholders, so from a commercial roadmap, development, and regulatory. I really didn't want to talk about classification. What I did want to talk about is certain things that small companies need to have in place, and those are small and medium-sized enterprise certification within the European Union and also within the UK as well. So if you are a small company or small or medium-sized enterprise, you need to get this certification in order to meet certain criteria for free use of certain pathways and certain tools that the European Medicines Agency utilise, or that they have designed. So you need to have this SME certification, so that it needs to be recognised by the European Medicines Agency, and you need to apply to the European Medicines Agency SME office. Also, any company that does any work regulatory procedure with the European Medicines Agency has to be registered on the OMS registration database. So that's the Organisation Management Services. In order to do anything, in order to submit a clinical trial application, you need to be registered. So that's one of the administrative duties that have to be carried out. I mean, I've taken out this and I've put the ATMP classification in another milestone. And that's the end of my presentation. I hope you've enjoyed it. If you've got any comments, please don't hesitate to leave them. This was a bit of a freestyle talk. I didn't really have any notes, but I have been doing a lot of work, and so I just thought I'll just quickly go through this because there's a lot more to come. Here are my references. Thank you so much. I hope to hear from you again next time. Bye-bye.